I think it's 1:30. We start the talk. My name is Feridun Hormozdiari. I'm a faculty in UC Davis, and the title of my talk is Genomic Variation and Genetic Module Discovery in Neurodevelopment Disorders. So based on the title of talk, it's obvious that I will be talking a lot about neurodevelopment disorders and autism and intellectual disability in the context of finding genetic variations which are contributing to these disorders, and also finding genetic modules that might contribute to these disorders. So the outline of talk is, first I will talk about general genetic variations that might be contributing to neurodevelopment disorders, and I specifically will be talking about structural variation discovery and method that we have developed for structural variation discovery and its application in understanding these sort of disorders. Then I will talk about methods that we have developed in how to combine different genetic variations that we have found in these disorders in the context of finding pathways and modules which might be important to these disorders. During the short talk, I realized that a colleague of mine has talked about some of these methods, so I will talk a little bit less about structural variation discovery, and instead I will also talk about the third method, that third project that is about early prediction of these development disorders using genetic variations. So let's start with some basics. When we compare different human genomes, there are different type of variations that we hope to observe. The simplest type is SNPs or SNVs, is that one base pair changes. For instance, the G changes to C. Another type of variations is indels. That's when a single or few base pairs are inserted or deleted between one genome versus another genome. And the third type of variation, which is a type that I work a lot, is structural variations. And that's defined as large rearrangements in one genome versus another one. And large, typically, historically, is defined anything above 50 base pairs. It's kind of based on technology. So any type of rearrangements that's bigger than 50 base pair is considered a type of structural variations. Different types of copy number variations fall in this category. So there is very known relationship between frequency of different variations and size of variant. We know that the most common type of variations that when you compare two genomes is single nucleate variants. As the size of variation grows, you expect to see less and less of them. And there are different ways to assay these variations. Traditionally, SNPs were the easiest one to assay. You use SNP genotyping technologies or Sanger sequencing. For copy number variation, we used to use array CGH. For very large CNV, there were other technologies. But now, with advent of next generation sequencing technologies, we can almost assay every type and every size of variations with quite a high throughput, as long as we can develop computational methods to analyze these data. So let's look at a little bit more into structural variations. What are different types of structural variations? For instance, a typical type of structural variation, let's assume this sequence is the donor genome or the new genome that we are analyzing. And this is the reference genome that we are comparing this new donor genome against it. One type of structural variation is indel. For instance, a sequence is inserted in the genome, in the donor genome versus the reference genome. It might be deletions, a chunk of genome is deleted. It might be inversions, when a chunk of genome is inverted. It might be different types of transpositions or translocations. There are when chunk of genome copies and cuts and pastes itself to another location in genome. All these different types, and you can imagine much more complex type of rearrangements too. These are all categorized as structural variation, and our goal is to develop computational methods which are able to discover these types of structural variation. And why do we care about this type of variation? Is that for many diseases, we know structural variation to be one of the major contributing factor. For instance, for a common type of leukemia, it's been known for a long time that a particular translocation is responsible for that type of cancer. Or in the disease that I study, autism and intellectual disability, there are many well-known structural variations which are contributing to this phenotype. For instance, a deletion or duplication in chromosome 16, known as 16p11.2, is known to be responsible for over 1% of all the autism cases. So these are different types of variations that we are trying to find using the methods that we are developing. So with advent of high throughput sequencing technology, the field of genomics has been revolutionized. If before it cost us around $1 billion to sequence one genome and produce the reference genome, now we can sequence tens of thousands of genomes with ease, and they are happening everywhere in the world. They're sequencing thousands of samples. And this is because the cost of sequencing has now dropped to around $1,000 per genome. And I'm sure you have seen this plot many, many times, but this wouldn't be an official genomics talk if I don't show this plot. So, but this is great. It means that we have a lot of power here. But let's not forget its drawbacks too. One of the biggest drawbacks that these technologies have is the read length produced by these technologies is quite small. Usually they tend to be somewhere between 75 base pair to 200 base pair based on the technology that we have. But for instance, in human genome, we know that 45% of human genome consists of repeat elements. These are different types of repeats that are all over the genome, has created a lot of complexity for analyzing these genomes. Combining the short reads with the repeat genome, you can see that it's a recipe for disaster for calling variations, especially structural variations. So how does a general variant calling at the moment work? It's the most technologies that we are using an approach known as resequencing. And the idea is that if we have a donor genome and a reference genome, majority of the genome is going to be identical. 99.9% is going to be identical. But those very small differences, those variations, that's what we want to find. So the approach that is working best right now is that when you sequence the genome, it might be targeted or the whole genome sequence. What you will do, if this is the donor genome that's been sequenced, and we don't have the official genome, we have just these reads produced, what we try to do is to map these small reads back to reference genome in order to find where officially they're coming from. So in other words, when we map them, and if we can find exact locations that they're coming from, then based on the discrepancy that we see, we can call variations. For instance, here, just by looking at, we say there are two locations that are different between the mapped reads and the reference genome, so there is a variant here. But there's some complexity here, and one is that you should trust what you have sequenced. If there is errors there, what you're calling might be just sequencing error. And the other one is that incorrect mapping, because I said the genome has a lot of repeats and also the sequence reads are quite short. If you're doing a mistake in mapping the reads to the correct location, then the predictions that you're making is going to be false. So this is a big and problematic issue. A little bit more about the technologies. The current technologies that produce high throughput sequencing technology, at least for the second generation sequencing technologies, provide what's known as paired-end sequencing. Let's look at it how this works. Is that you get the DNA genome, it's fragmented into small pieces. These small pieces is fed into a sequencer. The two ends of each fragment is sequenced, producing what's known as paired-end sequence. So each of these ends is somewhere between 75 base pair to 200 base pair based on technology, and this fragment length itself usually obeys a normal distribution or something like normal distribution. Usually the technician who produced the library might not do an amazing job, so it might not look as good as this, but that's what we are shooting for. And there is a minimum and maximum on the size of these fragments that they try to impose when they're doing sequencing. So the key point here is that the fragments kind of obeys this distribution. We have a delta min and delta max, which tells us what is the minimum value and maximum value of the insert size. And the two ends of these fragments have been sequenced and provided to us, and in between is not. And what we want to do is to use this sort of information to call structural variations. So the framework that has been developed is that, let's say this is the reference genome, and these are the reads that have been produced from sequencing the donor genome by the sequencer. When you map these reads back to reference genome, you expect, as I said, the two ends to be we know what the size that they should obey to look like. And when we do the mapping, if they obey what we expect, we call those reads concordant reads or concordant mapping. But if the size doesn't obey what we would expect it, we call that discordant reads. This shows some sort of discrepancy from what we would expect the reads to look when they're mapped from what is really looking. And this information can be used for calling structural variation. How? Let's say this is the donor genome, and let's say these are different structural variation in comparison to reference genome. Let's say there is an insertion in the donor genome, this green box is inserted. If you have a paired-end reach which spans this green box or the insertion, when it's mapped back to reference genome, you can see that it's going to map closer to each other than what's expected, because there's an extra sequence in the donor genome which does not exist in a reference genome. Or if there is a deletion in the donor genome, this red block is deleted in the donor genome. If you have a paired-end read, when it's mapped back to reference genome, the two ends are going to map much farther from each other than what's expected. Or if it's inversion, you would see a inverted, one orientation will be inverted. So this is a type of a signature that usually we use for calling structural variations. So our goal is to be able to develop computational methods which are capable of using this type of signatures for calling different types of structure variations. So I will try to summarize the methods that we developed in one slide. This usually is 10-minute talk, but I will try to summarize here because people have talked about this in the short course. So let's first consider we have a single sample. These are different reads that have been mapped discordantly. In other words, they show a type of structure variation. And because we want to consider repeat regions, each one can map to multiple locations. So for each of these reads, we have multiple locations that they map. And now we want to be able to call structure variation using these different mappings. One formulation that we came up was to try to minimize the number of structure variations predicted such that every read is assigned to only one location. And we call this problem maximum parsimony structure variation detection because we are minimizing the number of structure variations. So in other words, what we are trying to do is to assign every read, every discordant read to only one of the potential mappings that it can go to. In other words, assign uniquely every read to one location such that the total number of structure variations predicted is minimized. We showed that this problem is NP complete, and you think about it, you can use set cover reduction from set cover to prove this. And we provided an algorithm which we call Variation Hunter, which is a order log and approximation for this problem. It has two main steps. Step one, you try to group all the discordant read mappings into clusters, and then you try to pick the minimum number of them. But what we observed was that this was for single sample. When we had genome from single sample whole genome sequence and we mapped back to reference genome and we made the predictions. But when we have multiple samples, let's say members of the same family or members from the same population, when you use an approach like this and you make prediction for every sample separately and you compare them, what we observed was that there was a lot of differences between them that we weren't expecting. And that's because the drawback of the methods that use only one sample to call is that if they even make a mistake in one sample, not call the variation in one sample, then you're going to say there's a difference between these two samples, which in reality there was not. So the approach that we came up was, why not combine all this data that we have from multiple samples and try to do a simultaneous structure variation prediction in all of them? So instead of minimizing the number of structure variation, we minimize a weighted summation of structure variations picked. And again, the problem is, of course, NP complete, and we can give a logarithmic lower bound and approximation and give a method that achieves that approximation. So I'm not going to go into details of the method, but I would like to show one slide, which is, I think it's important. I was debating to show it or not. This is an extension of the method that we developed, Variation Hunter, and we ran it on two genomes, CHM1 and CHM13. I'm sure you guys are going to hear about these genomes more soon because it's becoming a standard for variation detection because we have PacBio data, long read data on these two samples, and we have very complete assemblies of these two genomes, so we can do comparison. And we ran our method TARDIS and two other methods for calling structure variation on these two samples, which we have truth set of structure variation. That's important. That for these two samples, we really know what are the structure variations. And we made prediction when we compare them. There is a high overlap between them. That's good. The false discovery is not that bad. I mean, you can around 20%, and you can filter more and bring it lower. Something around 10% is achievable. What is important is this part. Even combination of these three methods, union of these three methods, did not break the 50% true discovery rate. This means that for calling structure variation, even our best methods that we currently have, even union of them is not able to capture 50% of structure variations that we can capture using long read. This means that still, and this also holds for indels, even indels of size 5 base pair or 10 base pair. This means that all the methods that we see, we do whole genome sequencing and we do analysis and we call variation, especially for structure variation, we are missing 50% of structure variations easily. Yes?
So here the gold standard is long read data?
Yes, exactly. So we use the gold standard because for these two samples we have PacBio data, we can do a really good job in calling variation, but we still are missing, we know in PacBio we are missing variation too. The very long inversions or those deletion or duplication which exactly happen in the identical regions, those are still being missed. But even in those data, we can see that over 50-60% of the deletions and duplications and indels are not being picked by our current approaches. And I would like to say even this is simpler than most genomes. And these are, although they are human, these are haploid genomes. These are not diploid human genomes.
Did you try combining the two samples? You said earlier that it's going to call them
We did. So when you join the calling, of course, the depth of coverage goes higher, and it does a better job for the calls that absolutely agree, both agree, but not by far, a little bit. So what happens is that a lot of the things that we are missing, I mean, you would think a lot of things that we're missing is that we are picking the wrong mapping. We are picking the wrong mapping, but what happens is that it goes and maps perfectly in other location. So even they don't give us a discordant mapping for us to go and fix it. We're not even getting it because it maps perfectly to another location. We say, oh, that read is good, but it's not good. It has to go somewhere else. Another thing is that when there's a GC bias, when there is a very high GC, even some places are not being sequenced by Illumina technology. Okay. So any questions till now? Because I didn't go that much into details of the method because I want to talk a little bit about the results and go to the second part. If there's any questions about how these methods work, I would be happy to talk after the talk. So talking about autism and in general neurodevelopment disorders, here a small background. In general, development disabilities cover around 15% of the children. Autism spectrum disorder is somewhere around 1.5%, which is quite a high number if you think about it. There is a very strong genetic component for autism. The heritability is assumed to be above 50%. Now the number is the new papers are saying around 70%, which is really high if you think about it. There's a very interesting male to female bias, this is always kind of surprising for me, is that for every 4 boys who gets diagnosed with autism, only 1 female gets diagnosed with autism. The rate of autism in the past 10 years has increased, especially in US. It has almost doubled. It can go from very severe intellectual disability to very mild sort of Asperger syndrome. And our goal is to find all the potential genetic causes of autism and try to understand what causes this disorder. There are 2 patterns of inheritance that people study for autism. One is known as simplex family. That's when you have mother, father, and several children, and only one of the children is affected by autism. Other members of family are not. This is known as simplex family. And the other one is that several members of the family are affected by autism, and that's known as multiplex family. And all the results that I'm going to show you guys, it's on simplex family. The data on multiplex family is, at the moment at least, much fewer than simplex family. It's easier to analyze this sort of families. And what especially we are looking for are what we call de novo variation. De novo variations are a variation, can be structural variation, it can be any type of other variation, as long as it's not inherited from parents. These are brand new variations that happen in that sample, which potentially, if we are dealing with simplex family, these variations might be able to explain that disorder. So I would first like to show this result from this paper, which is very interesting, at least in my opinion, is that they sequence somewhere around 2,500 ASD families, that's mother, father, an unaffected child or sibling, an affected child or proband, and they tried to call de novo SNVs and indels, and these are whole exome sequencing. Only the coding regions have been sequenced. And what they observed was that there is very clear, significant enrichment of de novo mutation, SNVs or indels, hitting the probands, the ASD affected children, versus siblings. And you can see it very clearly on substitutions, on missense, and it becomes much stronger for loss of function variation. But you don't see that for synonymous. For synonymous, it's almost the same. So this shows that de novo variations, de novo coding variations, which changes the coding sequence, potentially has a significant contribution to this disorder. So this is a very key figure. If there's any question about this.
What is LGD?
LGD, loss of function mutations. These are like frame shift, stop gain, this sort of things. These are not clearly destroying the function of the gene, not necessarily any coding variation which is not missense.
I was going to ask the question, since this is known to be something that moves on a spectrum, and here you have balls and squares that are either black or white, right?
Yes.
How clearly can we really tell someone a case, that is someone affected?
That's a very good question. So the good thing about this data is that the proband and sibling both have been seen by a clinician. So a clinician has made a decision.
The same diagnosis.
They tried to make it as good as possible. However, in general, diagnosing of ASD is not as simple as you might think, and they're based on different clinicians, it might be different. So there are severities assigned to this. For instance, the intellectual disability ones are much easier, but the ones that have social deficit might be a little bit harder. So there's definitely a big spectrum here, but this is as clean as the data at the moment that we have the Simon Simplex collection data. So this was all about de novo coding variation. So in another paper, they also looked at rare inherited CNVs and SNVs. These are again coding. They are inherited, but they are rare, meaning that, and not the definition of rare that GWAS people use, that less than 5%. This rare means that it has not been reported in 1 kg, has not been seen in any family. It's private to that family. And if you see even those ones, you can see a significant enrichment in the proband versus siblings. So we know coding rare variations also are contributing to the disorder, even if they're inherited, as long as they're very rare. Yes?
Do they split inherited from mom and dad?
No, they don't. The more significant comes from the mother's side. The ones that seems to be affecting more seems to be coming from mother's side, not from the father's side. It's a very, very interesting observation. And kind of, I mean, there's a theory out there that this kind of might explain why you might see it. I mean, if you accept there is a female protective, then you can explain that why you see it in the disease more in the sons versus daughters. So this was all done in whole exome sequencing. So the result was all about coding data. But we were interested to see what is the contribution of non-coding variation. So as a pilot project, 56 trios and quads were picked. It means 56 families, and sequenced whole genome with 30x coverage. And these are the same 56 that we had whole exome sequence data. So what we did was that we got the data, we ran the mappings, we used the GATK for calling SNPs. And first thing that we observed, and we looked at the de novo SNPs. First thing that we observed was that the number of the de novo SNPs increases by age of the father. This is the plot, and this means that as the fathers are older, the paternal age increases, the number of the de novo SNPs that children will have increases. And this matches nicely with the risk factor of the older father older fathers correlating with higher rate of ASD. And this might explain that to a good extent. And this data has been replicated by many different groups. So we are at the moment very confident that age of the father is increasing the number of de novo mutation. We did not observe similar pattern for age of mother, but there are other groups that have observed slightly increase, but nothing as severe as this. So then we also used our methods because we were very interested in copy number variation, structure variation. So the methods that I presented, we applied it for finding structure variations, and we found a set of structure variation which I'm going to present. But before going further, who has heard of this debate between clinicians that whole exome sequencing is better or whole genome sequencing is better, and always this fight comes in Twitter? So it's always, I mean, every month that the paper comes out, people start to argue which one is a better way to go forward. So here is our take, is that let's look at the only coding variation. Let's see who does better job at calling coding variation, right? So this is for calling SNPs. This is the result this is validated data. This is the result from genome, and this is the result for exome. They look quite, I mean, you can say the genome is slightly better, but not much. But when you look at CNVs, these are again CNVs that affect coding sequence. They delete a coding exon. You can see that genome does by far better than exome. And I'm not counting UTRs, I'm calling coding sequence. So if your goal is to call all coding variation, it seems whole genome does a better job than whole exome. But of course, it's more expensive. It's 3 times more expensive. So going back to the samples that we analyzed, using the method that we developed, Common Land Variation Hunter, we were able to call somewhere around 1,700 deletions per child. Around 10 of those deletions were private. The false discovery was quite low, was around 10%. And as a matter of fact, we found one true de novo deletion, 6 kilobase per, which was only found by our method, and it deleted a UTR of a gene which potentially can be important, we don't know. But we also saw something interesting, and that was we saw there was a lot of samples which had rare inherited CNVs affecting genes that we think are important for ASD, inheriting from both parents. So it kind of makes us think that maybe there is another model that you might have multiple hits coming from different parents, from both parents, and affecting result. But this is not something that is statistically significant because we didn't have enough samples, and it's just one-off example. What was significant was that we observed there's a significant enrichment of de novo and private SNVs or SVs. These are non-coding SNVs and SVs, which affect DNA hypersensitive sites of known autism genes. Although the significance is low, is 0.03, but there is a signal for non-coding variations, de novo or rare non-coding variations having an effect on this disorder. And this was only on 56 quads. Now we have replicated the same analysis in around 520 quads, and similar pattern is observed. So we feel that there is a contribution, although not as much as contribution from coding de novo variation. Okay, so this was what I wanted to talk about, the first part of my talk, which is about discovery of structure variations and variation in neurodevelopment disorders. Is there any questions about this part before I go to? Yes. Yes. I think she was first. Go. Yeah.
So I was wondering, so did you mention that in your data the increase of the de novo rate as the father gets older actually explains the increased rate?
It matches to a good extent by what we know, but it doesn't mean that it's all of it. Definitely, probably it's not. Probably there are a lot of other factors involved. But both of those two are true. As fathers getting older, there is a higher rate of ASD, and as fathers getting older, there is a higher rate of de novo mutation, and we know de novo mutation is contributing to ASD. So there is a path there, but it's definitely does not mean that's a one-to-one. Not at all. Yes.
There's probably someone important.
Uh,
Oh, yes.
Sure.
Just following up on it. So this linear relationship between de novo mutations and fatherhood, that's also been observed in healthy.
Yes. This has been observed in exome sequencing, healthy, non-healthy. It's been reported in the past few years a lot. The only question is that is it linear or not? Other people have modeled it with different. Some people have the different models, but it's definitely increasing. It might not be linear.
So is kind of is the rate higher in the autism data or
Rate of de novo mutation. Rate of de novo mutation.
With age, say, is it stronger for mutations that potentially affect autism?
So, because the number of de novo coding mutation is very low, you're talking about one de novo coding mutation per sample. I can't say that, but I know that there is more de novo mutation in ASD patients that are changing the protein coding versus Provine. That I can say.
A related question, this phenomenon of increased with age, do we know if it's kind of uniform across the genome, or if it affects certain regions, certain loci?
That I don't know, but I know the rate of de novo mutation is not uniform or genome, but the increase I don't know, but rate of de novo mutation definitely it's not. There are hot spots. There was this paper by.
Yeah, I was wondering if this age phenomenon
That I don't know.
has a preferential area.
I don't know that. I don't think anybody knows that. I don't know that. Yes.
To follow up on this too, I was wondering if you, we know, even though the GWAS is not as successful yet for autism, but have you tried in that sample where you have a relationship between the polygenic risk score and see older fathers also have a higher risk? Because it could be a sort of a spurious thing you see. And coming back to whether the de novo rate explains mutation, you see if maybe older father.
That might be the case. I didn't check. That might be the case. And to say that I want to be clear, they're increasing the rate of de novo mutation. We don't know if it explains the whole thing or not. That I don't know. I just know these two are correct simultaneously. What is the relationship? I don't know. But no, I didn't do that. I think. Okay. So till now, what I talked about it was finding variation, including especially de novo and rare variations, and finding structure variation and trying to explain the disorder. But what is interesting is that what we found, especially when you look at the de novo mutation, there are hundreds and hundreds of risk loci. The number of genes that are currently implicated in ASD is over 500. Some people are suggesting around 1,000 genes are contributing to this disorder, and it's very hard to know. First of all, because of the lack of recurrence of the de novo mutation hitting the same gene, it's very rare to see de novo mutation in 2 different samples hitting the same gene unless the gene is really good. So it's affecting many genes in different people. And interpreting missense mutation is very challenging. For loss of function mutation is easier, but when it's missense mutation, it becomes very hard to know what its contribution is. So our goal in the second part was to see if we can find a way to unify all these de novo genetic mutations that we are seeing into finding pathways and modules which are potentially contributing to this disorder. So I would like to first give a definition of what I mean by a module. So the definition I kind of like. It says that a module is an entity composed of many types of interactions, especially molecules and protein or protein interactions, which has a specific function, which is different from other modules. So for finding such modules, there are different ways to do it. What I would like to consider is, one, I would like to consider protein interaction networks. I would like to say that a module is a set of genes or proteins that are very much interacting in protein interaction. And second, I like them to have similar expression profile. And what I mean by that is that when you look at the expression profile of 2 genes, if they're in the same module, I want to see a similar profile when they are, for instance, if you're looking at the brain development for neurodevelopment disorders. And I'm going to define a disease module, for instance, ASD module or neurodevelopment module, as a module, as I defined here, which is enriched in de novo mutation in cases. You see a lot of de novo mutation in cases, and you should not see a lot of severe mutation in that module in population. In other words, if you sequence a lot of normal people, you should not see a lot of severe mutation in that set of genes that you have picked, or that cannot be a disease module. Let's look at a toy example. Let's look at 3 genes of CHD8, TCF4, and beta-catenin, and all 3 are genes that we believe have been shown to be very important for neurodevelopment. They have been shown to be important for ASD. When we look at the time the number of the autism cases that we had, each one of them had 2 samples which had de novo mutation in each of these genes, which at the time was quite a lot. And then look at the exome data set of controls that we had from ESP. Remember, these are de novo mutation. These are not de novo. These are just severe mutation because we don't have for controls that many family sequence. And we see for each one of them there is no severe mutation in these 3 genes. Then look at the expression profile of these 3 genes during brain development, starting from early fetus till adulthood. And then because we have this data from Allen Brain Atlas, which I will explain a little bit more, we can calculate what is the correlation between the expression of any of these 2 genes. We can use just calculate the correlation coefficient and see that it looks quite good. They're correlated with each other. And then we can look at the protein interaction network and see these 3 genes are interacting with CHD, it's interacting beta-catenin, beta-catenin is interacting TCF4 in the PPI network that we have. So this kind of means, in this toy example, is what I'm looking for. I'm looking for signatures, looking for set of genes that look like this. And I want to develop a method to find sets like this. So to more formalize it a little bit more, I'm looking for finding a connected network in protein interaction network that satisfies these conditions. I want to see a lot of connections in protein interaction between the set of genes that I pick. I want the average co-expression between those genes to be high. I want the total number of severe mutations that I'm observing in this set of genes in the control population to, in general, to be very low. And I want to maximize the total number of the de novo mutation that I'm seeing in cases. So this is what I want to do. And it's easy to see this problem is going to become NP-complete. The moment that I say that I want it to be clickish for PPI network, I'm going to max click and the problem becomes NP-complete. But we can provide some heuristics to try to solve this, and that's what we did. We provide a heuristic which we call MAGI, short for Merge Affected Genes into Integrated Networks, and it's publicly available. I will try to give a very high view of how the method works. It looks at the protein interaction network for every gene, co-expression network, severe de novo mutation, loss of function or missense per gene in cases that we have in de novo in the probands that we have, and also severe mutations in control population. It calculates a score per gene, and then what we do is we try to find small paths of size 5 to 8 genes, in other small paths which are connected in protein interaction network, and they are significantly mutated in the cases, and the number of mutation in control is very low. And it sounds like an easy problem, but it's not a very easy problem. And the way that we solved it was using a technique which you guys heard yesterday Fabio talk about it, color coding. It's a very powerful technique which can be used for different problems. And what it does, it's very good at finding simple paths. If you're trying to find simple paths of size less than log n, this is pretty good. Especially if the function that you're trying to maximize is additive, which in our case is we want to maximize the number of the coding de novo mutation that we are observing in this path in cases. And I will tell you how we used it. Then after finding these paths, which we can find many of these paths because it's a randomized algorithm, we can run it many times and find many good paths. We try to merge paths using some random walk approach. We try to merge them and create a bigger network. And then we use a local search to throw a gene out and add another gene to improve the number of mutations that we cover. And we can run that many times till we have, because it's a randomized algorithm, we get many clusters. We pick the one that has the best score, and we try to merge the top 1% to create a bigger network. So this is high level how the method works. Is there any question? I would like to talk a little bit more about the color coding, although most of you guys are now quite familiar with it after Fabio's talk. Okay, so color coding, it's a very powerful technique, was developed in 1995 by Noga Alon et al. And it solved this problem. Given a graph, an undirected graph, it wanted to find a simple path of size k, with k is less than log n. And the way that they solved this problem, it was a randomized algorithm. They said, let's color this graph with k different colors. And then let's find a path in this graph, which is a simple path, and exactly uses these colors, each color exactly one. So if you can find such a path, you have found a path of size k. And then, because your coloring might be not very good, there might be a good path, but you color the two of the same nodes in the path, you might have colored it with the same color, so you will never pick it. So let's randomize this and apply this many times, 2 to the power of k, because k is logarithmic order n time, and see if we can find it then or not. And they can prove that with high probability, if you repeat this approach 2 to the power of k times, you will find, for once with high probability, one coloring, which is each node has colored one time for that simple path that you have. And when you have the coloring for finding the path, you can use a dynamic programming, which you can have for every vertex v and every substuff colors k. Let's call that substuff colors s. You can find a path which exactly uses that colors in s and ends at vertex v. Okay, so this was the general color coding that was used. And many papers have been published in the past few years that use this approach for different problems. It's very powerful. So what we wanted to do, we also want to find a paths of size k in the PPI network, which the number of mutation cases is maximized, but we have a second constant, the total number of mutation in controls is less than some constant j. And we assume this constant j is not big. So how we solve this, we just simple extensions to the color coding. First, in the matrix, in the dynamic programming matrix, instead of just having 0 or 1, we count the number of the de novo mutation in cases because it's additive that easily can be added to the dynamic programming. But for the controlling the number of mutation in controls, what we will do, we add the extra dimension of dynamic programming. So we have now, instead of a 2-dimensional table, we have a 3-dimensional dynamic programming, which the third dimension controls the number of the mutations observed in control. Yes?
What if you consider a relative measure instead of adding an extra dimension? Just, find the ones with high ratio of mutations in cases versus control.
So if you define the ratio for each node, if you divide the cases by the number of controls and you want to sum that up and make it maximize, that will work. But if you're doing it, summing the number of mutation in controls in all the path and dividing by the, then you can't do it. Okay. Do you see the difference?
Yeah.
For that one works, the other one it doesn't work. But because most of the controls mutation is 0, the whole thing becomes an issue here. Okay, so this is the general the extension that we did for color coding, and it works really well. It's quite helpful, finds really good paths for us, and then we try to merge the paths and find the modules that we are interested in. So then what we did, we applied, at a time we had a whole exome sequencing of around 1,000 or so cases. That number at the time was big. Now it's around 10,000 or so. So we applied our method for protein interaction networks. We used the combination of HBRD and string network. For co-expression network, we use the RNA-seq data from BrainSpan Atlas data. I just want to give a shout out to this data set. It's produced by Allen Brain Institute, and it's amazing data. If you're working on anything related to neurodevelopment, neuropsychiatric, anything that needs expression of genes in brain during fetus time for different regions, during time point, is really good data.
And it's human.
They have human, they have mouse, and I think they have one of the great apes. I think they have macaque, I'm not sure, but they have human and mouse, that I know. And the human data is very expensive, and it's publicly downloadable. The RNA-seq is not available, but the RPKMs is downloadable. So, but they have fetus, which is quite amazing for different regions of the brain. And for control, we had to use the exome sequencing project data, but now there is EXACT, which is by far larger, but at the time we didn't have that. So applying MAGI, this is the core of the module. This is the nodes that in any, because this was a randomized algorithm, we find many modules, but this was the set of genes that always were picked. The 3 genes that I showed you as toy example, beta-catenin, CHG8, and TCF4 have been picked. And the color here indicates the number of the de novo mutation in cases that have been observed. The more red it is, the more de novo mutation we had in that sample. If you see, there are a couple of genes which are gray, like EP300 or MYC, which we didn't have any de novo mutation in cases, but our method anyways picked it because it needed for connecting and satisfying the condition. This is the full network, how it looks. It was around 81 nodes. It was interesting that it was enriched in some of the genes that we know are important. For instance, for Swine-Soif complex, it was significantly enriched. The whole Swine-Soif complex was almost picked. The Wnt pathway was picked, a lot of the genes, and so on. Then we used the permutation test to see if this module, the number of de novo mutation observed in cases, was significantly enriched in the probands, and it was. Which was nice. But we also repeated the reverse. We said, let's look at the siblings, the ones that do not have the disease, and run the module and find it. And that one was not significant. The module that was found was not significant, which was again good. And interesting, as I said, the pathways that were very popping up was Notch pathway, cell cycle, Wnt pathway. And it's very interesting because these are not, especially cell cycle and Wnt pathway, are not something that, at least at a time, that people were looking were thinking that those are important for autism. But now this being replicated by other groups, we know chromatin remodelers are now very important, which is surprising. You would expect brain genes to pop up, but these are not brain genes. These are general development. They're expressed everywhere at every time point. So I presented a few years ago, when I was working, I presented this result to Ron Shamir. We were we were meeting, and he said, what happens if you delete this set of genes completely and redo your method? And that's exactly what we did. And then we found a second module, much smaller, but now these genes are very much brain genes. If you look at it, these are all expressed in brain. It's long-term potentiation, which is about how neurons work together. It's about calcium signaling, which we know it's very important for how neurons work. That was great. So we again showed that it's significant. And also, as I said, long-term potentiation was really high, and that's a very big function of the brain. So that was great. So we found 2 modules, which one was for general development, one was very specific for brain and how brain functions and how synapses communicate with each other. So going back to the Allen Brain Atlas that I mentioned, as I said, we have the expression for different genes in different regions of the brain, starting from early fetus, 8 weeks post-conception, going to birth and afterwards. So this is really in-depth data that we have. And we looked at the average expression of each of these genes in different time points. And the first module, which was for general development, we observed that it was really high at the beginning, and then as it became less, and especially dropped after birth. Well, the other one was for synapses communication, and we saw that at the beginning expression was lower, and then it becomes much, much more. That it shows a very different profile, the 2 modules. The expression profiles were quite different, which was interesting. Then we were interested to see, well, one of the reasons that we wanted to build the modules was to study missense mutations. So we said, how does the missense mutation? So there is this tool that exists, which is quite popular now, known as C-score or CAT score, which gives score to every mutation, especially missense mutation, of how destructive that mutation is. They use evolutionary profiles to find such scores. We said that let's look at the missense scores that we get for the missense mutations that are inside module versus outside module. So the blue ones are the scores for missense mutations that are inside modules for probands, the ones that have disease. Red ones are outside modules for probands which have disease, and green ones are missense mutations that scores for siblings and controls. As you can see, the siblings and controls and outside module kind of look similar, although the outside module for probands is slightly more. but the ones that are inside modules, the missense score that was assigned to them was much, much higher. So we believe this gives us additional evidence that the genes that we are picking is potentially important because the missense mutation in them have been scored higher. And another way we can use these modules to kind of come up with some scoring mechanism for missense mutations. But this Z-score is also a pretty cool data source if you want to find some sort of scoring for different mutations. There are other approaches right now available that people use. So finally, we were wondering that were we able to find any new ASD genes, and potentially the modules that we found were replicated by new studies that came out, because so many other whole exome sequences were produced in the past few years. What we observed was that, for instance, in the additional studies that was published, when you look at the modules that we have picked, they were eightfold enriched in de novo mutation in the genes that we picked, which was nice. But more importantly, if you remember the gray genes, the genes that we didn't have any de novo mutation at the time, now that we have a much larger study, we are seeing a lot of de novo mutation hitting those genes. For instance, a couple of them we are sure that are important for neurodevelopment. So this means that the method was able to pick some genes that can be important for neurodevelopment. And then there was a very large intellectual disability study published that even enrichment was higher for that study. So this was the second part of the talk I wanted to give, was about the module discovery. And any questions for this part? For the last part, I will go very fast because we don't have that much time. So any questions about, yes?
Maybe just for clarity, the result you showed when you found the module, so that was also based on the gene expression, the connectivity to CPI,
Yeah.
and then the mutations you observed, few cases the same.
Yes.
Right?
Mutations I've observed in cases and population, not necessarily. It's not case control.
Okay. And then so when you then go to the program and the sibling, those mutations you observed, those are independent from how you derive your modules, right?
These ones?
No, like in the slides when you then show the histogram or the distribution. Yeah.
No. So I reorder I randomly do the mystery, and then I see that it becomes significant or not.
Okay, and then so the mutations you see in your programs there, those were not contributing to how you assign your modules, right?
They were.
They were.
At the beginning, that's how I use the scoring. Then I randomly distribute mutation and see the score that I get before in comparison to after. How does it differ? So going to the last part of my talk, which I will just go very fast over because I think we're getting too close, is that we wanted to see if we can use these approaches for early prediction of ASD, because we can't find pathways, we can't find variation. How about making early prediction for ASD? And as you can imagine, being able to make early prediction of these disorders can be very, very important, especially for disorders for like intellectual disability or autism. It's been shown that very early interventions can be beneficial for the children, and even if you can do it prenatally, potentially give families different options. But there's some very big challenge here. One is that you want your method to be ultra accurate. You really don't want to make a mistake and say, go to a pregnant mother that, oh, your child is going to have ASD. Well, you're completely wrong. You definitely don't want to do this mistake. And on the other hand, yeah, we love genetics. We say it's very powerful, but we should agree that also it has its limitations. For instance, we know that for autism, genetic heritability is somewhere around 0.5, 0.6. It's good. It's not perfect, so there are stuff that we're going to miss. So I'm going to make two assumptions here. One is that using coding genetic variation, we can only explain a fraction of cases. We cannot explain all cases. All the cases that we see, we won't be able to explain using genetic variation. And two, just because a sample doesn't have any genetic variation that we think is causative doesn't mean it's not going to develop any sort of neurodevelopmental disorder. There's epigenetics, there is environment, there is so many other factors. So putting these two assumptions together, we're going to define an ultra-accurate complex disorder prediction model as follows. We want to be able to predict the neurodevelopment disorders only for a subset of cases very accurately, and for other cases and controls, we don't want to make any prediction. So what we want to really be able to do is to be able to say for a subset of cases, make prediction, these cases will be cases, and we won't make any predictions, and we don't want to make by any mistakes of calling a control as a case, and we won't make any predictions for other samples. So
for the other samples, you say
And
not say you're
exactly,
not.
exactly. So this kind of summary. Let's say blues are the cases and reds are the control. What I want to be able to do is to do this
Yeah.
very accurately. I don't want to make any mistake, assign a red to a blue. But the rest that I can't say anything, I can't say anything. And the reason that this makes sense is that genetic data will not be able to give me full, especially coding variation, to give me the full information. So what we do is that we assume we currently are considering only loss of function mutation, very severe mutations, and we assume for every sample I, a vector Si, which for every gene there's a value 0 or 1, meaning that gene has been knocked out or not, or there's a severe mutation in that gene or not. We also assume a gene similarity matrix, which tells us for every 2 gene I and J how similar their functionality is. I'm going to use the co-expression data that I have because for brain disorders that makes perfect sense. And then you can come up with a function to convert these vectors to a new vector, which assumes the gene similarity. What I do is I just multiply this vector by this matrix, but you can come up with a way cooler stuff to do it. And the output that I'm looking is exactly the model that I mentioned. To be able to make prediction for a new sample based on this genetic variation, it's the case or not. It's the case or no prediction. So we develop a method which we call ODEN, and it has 2 steps. Step 1, it tries to it makes a prediction of a new case to be a case if it satisfies 2 conditions. 1, it should be far from any other controls. If it's close to any control, it will not make a prediction. And it should be grouped with a lot of other cases after dimensional reductionality. And I will say what exactly I mean here. What I want to do in the second part is to be able to find a dimensional reductionality such that all the cases are grouped together, not all, a lot of cases are grouped together and no control is in that cluster. So in other words, if I have a set of red points in n dimension and a set of blue points, which are the cases in n dimension, I want to find a case subset which is by far smaller than n, a center point C and a radius R. In other words, I want to find a sphere after dimensional reductionality that a lot of blue points or cases fall inside sphere and no red point falls inside that sphere.
What the dimensions
Are the genes. Dimensions are the genes that after multiplication to the matrix. But so you can assume it's a kind of a yeah, exactly. Yeah.
Of the gene expression.
Gene co-expressions. And for the distance, I'm using a weighted L1 distance because I want to be able to weight each dimension such that I can create these clusters. For complexity-wise, we showed that solving such a problem is NP-complete. We use the reduction from equal subset sum problem. I'm not going to go into this proof. I'll just show you how the method general works. It first assigns a weight to each dimension. It starts with all genes having weight 1. It tries to find, given these weights, tries to find a center point C and a radius R, which covers a lot of blue points and no red point is covered. Then, assuming that it has covered these sets, tries now to find a new set of weights such that these blue points get closer to the center and red points farther. So I'll use a linear programming for doing this. It has 2 parts. First part tries to reduce the distance from all the blue points selected from previous step to the center and increase the distance from all red points to the blue points. And this, because of the linear programming, we can solve it. Then we can give the weights the first step and iterate and hopefully converge. We didn't prove it converged, but in practice, it does converge. So I just want to show one result is that running this on the data that we had, and I compared it against KNN, SVM, and general linear model with Lasso and ElasticNet. There's a lot of other models that we can compare. I didn't do that, but we compared for different values of K for K nearest neighbor, and for very low FDRs, we can see that we are doing somewhat better. Although, if you look at it, it's only 8%. So you might say 8% is a true discovery of 8% is not that great, but it's as good as we can do right now. Yes.
Did you just pick one ball or did you try to fit multiple?
I'm fitting only one ball. I tried to extend it to multiple ball, did not improve that much. Okay, so I just want to show. Yes.
What happens if you, because this, of course, could be noise, right? In the gene expression data that calculates into noise in the correlation, the covariation. Did you try to see what happens if you try to do some reduction on the gene expression data at the beginning to sort of denoise it? Granted that you say you're using very good gene expression.
No, we didn't try to any. we didn't try any like that.
I mean, it kind of changes the dimension of your program
Yeah.
to start with, because you would go to
Small,
PCA.
yeah, sure, we didn't try and fit, but
it
that's
might
definitely,
be
it might,
something useful.
yeah, use for instance something like PCA to do a first dimension reduction. I'm going to try, I didn't, but yeah, that is very much possible, yeah. One thing I want to show is that, so these are all the samples that we had from Simon simplex collection. This is their IQ. These are all the samples with some simplex collection which had loss of function de novo mutation. And these are the IQ of samples that our method, Odin, was able to correctly predict. And as you can see, the IQ is significantly lower for the ones that we were able to accurately predict. So this kind of tells me that our model was able to more predict the ones that were more severely affected. Kind of makes sense. Probably we are doing a better job at the ones that are more severely affected, probably because it's more obvious to pick them. Final slide is that I told you that there's a ratio of female to male. There's a significant difference between ASD. So here's the whole data. The number of female to male is somewhere around 0.15. So by far, we have more males in our data sets than female. The ones that have loss of function mutation, the difference is slightly less. But the ones that we were able to predict, it's almost, the difference becomes by far less. So this means that, again, for the very severely affected, the difference between female and male seems to be, at least for our model, to be gone. So it seems the more difference is on the less severely affected, or at least our model is less biased in that regard. The genes that were mostly expressed, the regions that the genes that were affected were mostly expressed was in early fetal development and early mid-fetal. They were not affected during adulthood. So whatever happened at very early, it seems to. So yeah, I'm out of time. So in conclusion, I talked about genetic contribution of different genetic variation contribution to ASD, especially structural variations. Current estimate is that various de novo variation, including loss of function, missense, CNVs, is contributing something around 25 to 30% of ASD. Other people's might completely, especially if you talk with the GWAS people, completely disagree. But this is what I'm seeing. The pathways that are very important include Wnt pathway, chromatin remodelers, long-term potentiation, RNA signaling, and I believe that we can start to make early prediction for some of these samples using genetic variation. It's going to be small. It's going to be 8%, 10%, but I think for that we are seeing that we can do that. So finally, I would like to acknowledge my PhD PI and postdoc PI, during which some of these works happened during that time. Also my collaborators and my postdoc who did a lot of work for the Odin. We are currently trying to hire strong computational people, and I would be happy to take any more questions. Perfect.
I was very curious if you could elaborate a little bit more about this expression data that you were talking about. So it's got, for instance, this time courses over development.
Over
Are
development.
they replicated over several samples, or is
They are replicated over at least 5 or 6 different samples, and now it's more even, yes.
And they give you data by sample?
They gave you data by sample, by subtissue, by time points, yes.
Okay.
I think the number of things is around 450 different points that you have.
And are there also sort of variants annotated in this data? You know, the sample belongs to a person that blah, 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 blah.
No, you just have the age, you have the sex, that's it.
Okay, and the age means the development.
Well, because this is brain, right? That person had died at that point that they got the brain.
Okay.
I think that's it, nothing more.
Maybe
Because
there's a bit more for the mouse.
yeah, for mouse, I bet there is. I didn't play with it, but a lot more for sure, for sure.
All right.
Yeah.
Very important. Thank you.
Any more questions? Great, thank you.